Plants and their pollinating insects have evolved in such a way that size of their interacting parts matches closely. Size matching can be considered as an important driver of plant-pollinator interaction. The size matching story is more complex when it is considered in the context of the community, where plant species of diverse flower sizes and insects of various body sizes are found. Regardless of their size, insects can potentially visit a wide variety of flowering species in the community. However, they preferentially forage on flowers whose nectar tube matches their tongue length. So, what is the reason for the high preference and frequency of size matching interactions? Do they have functional advantages for the mutual success of plants and pollinators? To answer this question, we studied plant-pollinator interactions in a community of Lamiaceae. Lamiaceae family presents an ideal system for such a study given the high variation in flower size and a considerable diversity of insect visitors with a wide range of tongue length. We found that 3 types of interactions are possible. In type 1, the flowers are visited by insects whose proboscis length is smaller than the flower's nectar tube length. The insect has difficulties in accessing nectar, which can increase handling time and decrease the sugar extraction rate. The foraging efficiency is therefore relatively low. At the same time, the chance of contact of reproductive organs with the pollinator's body is also low. That decreases the pollen transfer efficiency and thereby the pollination efficiency. In type 2 of interactions, the proboscis is considerably longer than the flower's nectar tube. The insect can easily access nectar and experience relatively low handling time and high sugar extraction rate. However, due to the lack of proper landing platform, the insect has to hover while feeding on the flower. This can significantly increase the energy costs of the visit and thereby lower foraging efficiency. On the other hand, the pollination efficiency is also low due to the lack of appropriate contact with reproductive organs. In type 3 of interactions, the proboscis length of the insect corresponds to the nectar tube length of the flower. Such size matching facilitates nectar access, handling, and extraction, thereby increases foraging efficiency. At the same time, the proper feeding of the insect within the flower increases the chance of contact with reproductive organs and thereby increases pollination efficiency. While all sorts of interactions were possible in the studied community, size matching interactions occurred more frequently than mismatching interactions. Such a tendency can be explained by the higher energy gain and lower energy expenditure of size matching interactions. Size matching index can be therefore used as a reliable metric in plant-pollinator network studies to predict the frequency of interactions as well as the effectiveness of visits in terms of foraging efficiency and pollination efficiency.